Okay, class, we're back now and we're looking at ligand-gated channels. Ligand-gated channels. Now, where this comes in is to explain the neuromuscular junction, but not only that, to even explain neuronal junction. See, a neuromuscular junction is a neuron hooked to a muscle, but also it explains neuronal junctions, a neuron hooked to another neuron. Neuron hooked to another. So it's a generic talk. It's a generic talk. Okay? So, a ligand is any substance that can bind to a receptor. It's a generic term. One type of ligand we will discuss is a neurotransmitter substance. Okay? That's a chemical. Ligand-gated channels oftentimes work to produce graded potentials. Now, what a graded potential is, the action potential was all or none. A graded potential is you give a little bit, you get a little bit. You give a little bit more, you get a little bit more. That's what we call, that would be a graded potential, okay? To produce graded, rather than the all or none action potential. A graded potential opens according to the magnitude of the signal. The stronger the signal, or the stimulus if you want to call it, the more and longer the gate opens, the ligand gate. Gated channels in neurons are generally excited. Gated channels in neurons, and I should say, generally produce. Let me change that. Produce. That's what I should have said. Gated channels in neurons generally produce EPSPs, which are excitatory postsynaptic potentials, and IPSPs, inhibitory. In skeletal muscle, a MEP is created, known as a motor end plate potential. Okay? So what I'm saying is these gated channels, ligand-gated channels in neurons can do an EPSP or an IPSP, and in muscle, skeletal muscle now. Skeletal muscle, a MEP, motor end plate potential. Okay, let's go a little further. There are many neurotransmitter substances. Each of them causes the opening of or closing of certain ligand-gated channels. The one involved in skeletal muscle is acetylcholine. That is the name of the neurotransmitter that turns on your skeletal muscle. You need that. If you don't have that, then your skeletal muscles won't work. And when we get into pathology, which I'm not doing today, get into pathology, we have conditions, myasthenia gravis, different conditions that could give muscle weakness due to not having enough of this. Okay? In order to discuss neurotransmitters, we must discuss chemical synapses. Okay, that's where we're going now. A synapse is an anatomical structure where a neuron connects to another neuron or muscle or gland. That's a synapse. A synapse anatomical structure where a neuron will either form a neuromuscular junction or a neuronal junction or a neuroglandular junction, because neurons do turn on glands. The two types of synapses neurons do are chemical synapse and an electrical synapse. The chemical is the one we're going to talk about, and it's the far most common. The least common synapse is the electrical synapse, which involves one neuron or muscle cell hooking together by gap junctions. Remember we discussed gap junctions in the chapter 4 stuff? Gap junctions involve electrolytes moving freely between one cell and another. The electrolytes activate the next cell. We'll talk about that. I'll talk about that when we do in ANP2, particularly when we do the heart. All right. Let's now look at the general anatomy of a chemical synapse. Okay. Here would be one cell. Here's another. In this case, this is a neuronal. Okay. Okay, example of neuronal. Let's put it there. Example of neuron. Okay, here's one neuron hooked to another. This right here is where the signal is coming into initially. So this is going to be called, this neuron's membrane is going to be called the presynaptic membrane because it's before the synaptic cleft. Now I want you to read on this in the book and other places. This membrane, which is the second one, is the postsynaptic membrane. Right here you have a cleft with extracellular fluid there, known as the synaptic cleft. All right. In the generic situation, in the presynaptic, this right here is called the synaptic end bulb. The synaptic end bulb. Let me show you how that would be. Let me find something here if I can. Let's look for a neuron. Okay, here's a neuron. See right here, this would be what I'm talking about. These terminal branches, when we get to neurons, have a swelling right there. And that swelling is called the synaptic end bulb. That swelling is called, now they call it here the bouton, which they can in England and other areas. But we also call it a synaptic end bulb. Synaptic end bulb right there, which would be right here if we look at the end of this axon. Okay? That swelling. All right, so we come back to this. See, here would be here coming down. Those would be these bulbs right there, synaptic end bulbs on that drawing. So here we are. Okay, so in this presynaptic neuron, you have these things we're going to call synaptic vesicles. Remember vesicle from the cell chapter? And vesicles contain chemicals. In this case, these vesicles contain neurotransmitter substances. These vesicles contain neurotransmitter substances in there, right there. And these contain the substances that must be released into this cleft to activate the receptors to do what we need to do, to open those ligand-gated channels. Okay? So I want you to study this and look at the drawings in the book. Okay, note the presynaptic membrane, the cell membrane before the synaptic cleft. The synaptic cleft is the space between the membranes. The neurotransmitter substance must cross this cleft. The postsynaptic membrane is after the synaptic cleft. The synaptic vesicles contain neurotransmitter substances and are in the presynaptic cell. The postsynaptic membrane has receptors for the neurotransmitter, and it has the ligand-gated channels. Okay? So we go further. How does the chemical synapse work? Let's talk about it. Action potentials. See, the thing you have to do is you have to have action potentials. So action potentials would come down here. Now think of this as three-dimensional. Action potentials will come down here, and they're going to come all along this. Now, I'm going to throw something in on you here. Action potentials coming down 3D in all the directions, coming down 3D, eventually are going to meet one another. Now, the question I often ask if this was class, they're going to meet one another, because if they're coming down, how are they going to not? Then the question I always ask in class, when they meet one another, will they stop or keep going back up? Let me give you a quick answer since we're not faced. They will stop, because the action potentials coming this way, when they meet this way, they can't go behind that area of the membrane. Why? Previous subject, that membrane is in the absolute refractory period. So they'd have to stop. If they didn't stop, they'd go back up and you'd never turn this neuron off. So they'll come down, but they got to stop because of that absolute refractory period. Okay, but anyway, let's go further. They come down, and what they do is, now right in here, what you don't see, are voltage-dependent calcium gates. Now, I never brought this up before. Right in here, but in the text they will. Voltage-dependent calcium gates. So right up in here, what do we have? We have voltage-dependent calcium gates. We have the usual voltage-dependent sodium and potassium gates, because we wouldn't be able to do the action potentials. So the voltage of the action potentials opens up this voltage-dependent calcium channels, which are only right in this area. Calcium is an ion in higher concentration outside cells than inside. So there's higher calcium in this fluid here. The calcium then, once you open up these channels here, will come into here. And in several ways, that a little more detail that I want to discuss with you all, the calcium will cause these synaptic vesicles to be pulled to the surface. This calcium rushing in will cause the synaptic vesicles to be pulled to the surface. You can read it. Then when they pull to the surface, these vesicles fuse. And then they open up. And by a process we've heard before, exocytosis, they're going to release their neurotransmitter substance into here. Now, when you release it, you got fluid just swinging by, running, this extracellular fluid. Some of it's going to get whisked away, but some of it's going to get pulled to the receptors. Now, the receptors have specificity and affinity. See, you heard those two words before when I talked about enzymes, but it's a different scenario. They have specificity and affinity. The specificity is that they only will attach to the proper chemical, the proper ligand. They will only attach to the proper ligand. Affinity means drawing power. They will literally draw some of this stuff across. So not all of it will get washed away. Not all of it will get washed away. When the chemical attaches to the receptor, then it will open up ligand-gated channels. Ligand-gated channels. Ligand-gated channels. Okay, this particular neurotransmitter substance. Okay, so we're going to go, see, that's what's here. We're going further. Okay, now, when you do that, what are we going to cause? Well, now that's where we get into this. What happens then depends on the specific neurotransmitter substance and the specific receptors. EPSP, IPSPs, or MEPs could be formed. Okay? Now, EPSP is what I mentioned in neurons. Okay? So, an EPSP is not an action potential. See, it's going to occur up in this area. It is a graded local potential caused by the opening of ligand-dependent gates to sodium. See, the sodium comes in, so the gate would allow sodium to come in. What does sodium do? Make the inside more positive. See, right here, making it more positive. What would that do? If enough sodium came in, you may cause this neuron to do an action potential. Remember, these are ligand-gated channels. Around in here would be voltage dependent. So I need to bring in enough sodium to get voltage dependent channels to start up. Okay? So the EPSP is a graded thing. The sodium through the ligand gated, making the more positive. The amount of gates opening is due to the amount of stimuli. See, that's a graded. Thus a graded, unlike the all or none of action. Various substances, hormones that can cause the opening. The purpose of the EPSP is to bring the prosynaptic membrane either to threshold voltage or closer in order to initiate action potentials in the postsynaptic neuron. Okay? Now, we go to this other one. Some neurotransmitters cause an IPSP. You can read this. An IPSP's job is to bring it further from threshold. Because, see, what happens is, any neuron has when we get into nerves, which we haven't done yet, any neuron has a bunch of other neurons hooking to it. And so it's like a voting action. And I'll talk more when I get into the neuron chapter. But we have two things, an EPSP and an IPSP. An EPSP and an IPSP. Okay? Now, I'm going to close this now. And we're going to start another one because we want to talk a little bit extensively on the motor end plate potential. The motor end plate. This would be the one that's happening in the skeletal muscle. So we're going to do another video.